Hello, I'm Vittoria. I'm ending my third year PhD in Naples, and my research project is about multicellular implementation of PID controllers for robust regulation of biological processes. And as all of you know, synthetic biology combines engineering principles to endow cells with new functionalities by developing some synthetic gene networks. And this finds application in different fields, ranging from industry to environmental field to medical field. But as you know, introducing these circuits into cells can cause some undesired effects due to the stochastic and nonlinear nature of biological processes. And so cybernetics comes in help, combining synthetic biology with control theory to improve stability, robustness, and performance of these systems. And in particular, the most used architecture is the negative feedback, in which a controller compares the output of the process with the reference, and based on the error, computes the control action. And an essential feature we want to achieve is robust perfect adaptation, that is the property to maintain a stable and predictable behavior under context variability and perturbation. And in particular, control theoretic literature abounds with high-performance, robust control solutions, such as nonlinear controllers or discontinuous controllers. However, dealing with biological parts limits the complexity of this design because we have a limited availability of biological components that can be used. And so biomolecular integral controllers have gained traction in this context for their simplicity in implementation and also and especially for their ability to guarantee robust regulation. In literature, this kind of controller, as I think you know, has been implemented with the antithetic motif, which is made by two molecules that bind and neutralize each other, and that has been demonstrated to perform as an integral controller. In some applications, however, it is desirable to avoid oscillations and excessive overshoots, and so we can complement this integral action with a proportional and derivative actions. Indeed, the integral action alone can cause excessive oscillations, can slow the response, while adding especially the derivative action can improve the transient response by fastening it and also by reducing the oscillations and the overshoots. The problem is that this approach that we find in literature is an embedded approach, which means that the process to control and the controllers are all embedded within the same cells. And this can cause lack of modularity, which means we need to redesign the system if we change something in the control strategy, to have a metabolic burden on those cells. And also some cells are not accessible, so we cannot modify them endowing them with the synthetic networks. And so the literature proposes to divide the labor over a microbial consortium by embedding the process to control and the control parts across different populations in a microbial consortium. This can improve modularity, reduce the metabolic load, and also separate incompatible functions. However, multicellular approach also introduces other challenges, which are, for example, the increased complexity of the system or the need to manage the population imbalances, because in the presence of different cell populations, one of them will grow faster than the others. And also the need for introducing a communication mechanism to let the different populations to exchange signals. In these designs, the most used communication mechanism is the quorum sensing. Imagine to have different cells. These cells produce molecules. These molecules diffuse towards the external environment, and at the same time, cells divide and produce more molecules. At a certain point, the quorum is reached, which means these molecules are able to diffuse in other cells of the populations and also to produce an effect on them. And in particular, when a molecule enters the cell membrane, it binds to a regulator protein. This complex in turn binds to a target gene and modifies its expression for the desired application. So, combining the potential of biomolecular PID controllers with the advantages of the multicellular approach, the main goal of the research was to implement different architectures of the proportional integral derivative control family in microbial consortia. And in particular, what I've done is to design and validate in silico different controllers belonging to the PID family with this multicellular approach. In particular, all of these architectures contains a target population which embed the process to control, and then different controllers population which each one implementing one of the control action. The communication is made by means of quorum sensing molecules. In particular, the controllers, based on the difference between a reference signal and the sensor signal, produce a molecule which we can call control molecule that serves as actuating signal for the targets. And then the targets produce the sensor molecule that provide their states to the controllers and closes the feedback loop. In this presentation, I will focus only on the most complete architecture, which is the PID, because thanks to the modularity of this approach, we can derive all the other architectures starting from this one. This is the schematic implementation of the PID controller. So as I said, we have the target population embedding the process to control, which we assume to be made by two genes, X1 and XC. XC is directly affected by X1, and X1 is directly affected by the control molecule QU, and so by the control action. These targets produce the sensor molecule QX, which diffuse in the controllers, and these controllers contain different synthetic gene networks, which has been demonstrated to perform as proportional, integral, and derivative controllers. And so they compare this sensor molecule with the reference signal, and based on the error, they produce their contribution to the control molecule QU, which influences the target at the end. So the system can be described with a set of differential equations, but it is a real complex system. It is made by 16 differential equations, each one describing the dynamics of a species inside this architecture. And so I will show you directly the reduced order model, which is derived by making some simplifying assumption on the dynamics of the controller networks and on the dynamics of the quorum setting. In particular, we performed some timescale separation between the controllers and the process to control, obtaining this simplified model in which you can see the dynamics of the target process X1, XC, and you can see that X1 is directly affected by the control molecule QU, and in particular, the steady state value of this molecule contains the sum of three terms, which are nonlinear terms, but that can be demonstrated to perform like proportional, integral, and derivative actions, and beta P, beta I, and beta D are production parameters which play the role of control gains. Now, based on these reduced order dynamics, we performed some analysis by applying the classical control theory methods. In particular, we used a method that is called root contours. I will not go into details, but we applied the same design methods to all the control architectures developed, so the proportional, PD, PI, and PID. And we performed some in silico validation using, first of all, MATLAB. In these simulations, you can see the results of different simulations using the control architectures, where the target parameters were varied to account for the cell variability. And in particular, you can see that as in the classical PID controllers, when including the integral cells, the response is robustly regulated to the desired value, while this not happens with the P and PD controllers. However, the derivative action can modulate this transient response by reducing the overshoot and reducing the settling time. Next, we wanted to assess the effectiveness and robustness of the control strategy obviously, the final goal is the in vivo realization of these architectures, but the first step is to use a realistic simulator to perform some in silico experiments. And in particular, we used BSim, which is an agent-based simulator that is designed for bacterial populations and that allows to take into account the diffusion of molecules, the growth and division of cells, and other realistic effects. Here, for example, you have a simulation that I do not perform, but that explains how BSim can make some realistic simulations of realistic experiments of, for example, genetic oscillators. What we have done in BSim is to test the robustness of the control architectures to the two main factors, which are the cell-to-cell variability, that are differences in the expression levels among individual cells, and that are caused by uneven distribution of genetic material. And to perform these simulations, we assigned different parameters to daughter cells each time they split into two daughter cells. And next, we tested the population imbalances that are different relative numbers of cell types within the populations that are due to asymmetries in the metabolic load that make one population grow faster than the others. And we assign a different relative number of cells across the populations, in this case, neglecting the cell division in order to have a precise control over the number of cells in each population. And these are some examples of results. So for cell-to-cell variability, we performed a different simulation with different coefficient of variations of these parameters. And what we obtained is that all the control architectures are robust to this effect. And for the population imbalances, these simulations shows with the shaded areas the simulations that have been considered robust. And as you can see, adding the integral cells guarantees more robustness to population imbalances than the P and PD controllers. So in conclusion, we presented different multicellular architectures implementing the biological equivalent of the classical PID controllers. We designed the consortium, and in particular, we provide some guidelines to choose the control gains. Next, we discovered that the integral controllers can guarantee a robust asymptotic regulation, while the derivative action can enhance the transient response by reducing the settling time and dampening the oscillations. For future work, there are still some challenges. For example, we want to understand how to manage the population imbalances. We want to understand the effective effects of the sharing of resources within single cells. And also we need to carefully select the components to implement these strategies in vivo in the future. And so the future work will focus on these aspects. Thank you. These are my collaborators, Davide Fiore, Davide Salzano, and Professor Mario Bernardo, which is my supervisor. Thank you.